Welcome to the BioTech Whisperer channel. Our topic today is on the introduction to protein structure. If you are new here, we are a group of retired professors sharing our knowledge in bite-sized videos. Do subscribe and follow our channel for more updates. Let's dive into the topic for today. What are proteins and what do they do? Proteins are large, complex molecules that play many critical roles in the body. They do most of the work in cells and are required for the structure, function, and regulation of the body's tissues and organs. Proteins are made up of hundreds or thousands of smaller units, called amino acids, which are attached to one another in long chains. There are 20 different types of amino acids that can be combined to make a protein. The sequence of amino acids determines each protein's unique three-dimensional structure and its specific function. Amino acids are coded by combinations of three DNA building blocks, termed as nucleotides, determined by the sequence of genes. Within the cell, proteins are considered as workhorses and carry out most cellular functions. Proteins can be described according to their large range of functions in the body. Examples of roles protein play include the role of antibody, enzymes, messengers, as structural component or as transport or storage. For instance, messenger proteins, such as some types of hormones, transmit signals to coordinate biological processes between different cells, tissues, and organs. Knowledge of protein structure is essential to understand the behavior and functions of specific proteins. Proteins are polypeptides, formed by joining amino acids together via peptide bonds. The folding of a polypeptide can be described by rotational angles around the main chain bonds, such as phi and psi angles. The degree of rotation depends on the preferred protein conformation. Allowable phi and psi angles in a protein can be specified in a Ramachandran plot. A Ramachandran plot can be used in two somewhat different ways. One is to show in Curie which values, or conformations, of the phi and psi angles are possible for an amino acid residue in a protein. The second use is to show the empirical distribution of data points observed in a single structure in usage for structure validation. It provides for the probability of the theoretically favored region. Protein structures can be organized into 4 levels of hierarchies with increasing complexity. These levels are primary structure, secondary structure, tertiary structure, and quaternary structure. A linear amino acid sequence of a protein is the primary structure. This is the simplest level with amino acid residues linked together through peptide bonds. The next level up is the secondary structure, defined as the local conformation of a peptide chain. The secondary structure is the repeated main chain conformation, which includes alpha helices and beta sheets. The secondary structure is characterized by highly regular and repeated arrangement of amino acid residues, stabilized by hydrogen bonds between main chain atoms of the carbonyl group and the amide group of different residues. The level above the secondary structure is the tertiary structure, which is the three-dimensional complex arrangement of various secondary structural elements in connecting regions. This often involves multiple polypeptide chains. With that, the tertiary structure can be described as the complete three-dimensional assembly of all amino acids of a single polypeptide chain. Protein structures are stabilized by electrostatic interactions, hydrogen bonds, and van der Waals interactions. Proteins can be classified as being soluble globular proteins or integral membrane proteins, whose structures vary tremendously. Beyond the tertiary structure is the quaternary structure, which refers to the association of several polypeptide chains into a protein complex, which is maintained by non-covalent interactions. In such a complex, individual polypeptide chains are called monomers or subunits. Intermediate, between secondary and tertiary structures, a level of supersecondary structure is often used, which is defined as 2 or 3 secondary structural elements forming a unique functional domain, a recurring structural pattern, conserved in evolution. Stabilizing forces are critical to support these protein structures. In fact, protein structures, from secondary to quaternary, are maintained by non-covalent forces. These include electrostatic interactions, van der Waals forces, and hydrogen bonding. Electrostatic interactions are a significant stabilizing force in a protein structure. They occur when excess negative charges in one region are neutralized by positive charges in another region. The result is the formation of salt bridges between oppositely charged residues. The electrostatic interactions can function within a relatively long range, which is typically around 15 angstroms. Van der Waals forces is a general term used to define the attraction of intermolecular forces between molecules. There are two kinds of van der Waals forces: weak London dispersion forces and stronger dipole-dipole forces. These forces are instantaneous interactions between atoms when they become transient dipoles. A transient dipole can induce another transient dipole nearby. The dipoles of the two atoms can be reversed a moment later. The oscillating dipoles result in an attractive force. The dipole-dipole forces are more relevant for proteins as they occur in molecules that are permanently polar versus momentarily polar. In this type of intermolecular interaction, a polar molecule such as water attracts the positive end of another polar molecule with its negative end of its dipole. We call this attraction between these two molecules as the dipole-dipole force. From our current understanding, the van der Waals interactions are weaker than electrostatic and hydrogen bonds and thus only have a secondary effect on the protein structure. Hydrogen bonds are a particular type of electrostatic interactions similar to dipole-dipole interactions involving hydrogen from one residue and oxygen from another. Hydrogen bonds can occur between main chain atoms as well as side chain atoms. Hydrogen from the hydrogen bond donor group, such as the amine group, is slightly positively charged, whereas oxygen from the hydrogen bond acceptor group, such as the carbonyl group, is slightly negatively charged. When they come within a close distance, a partial bond is formed between them, resulting in a hydrogen bond. Hydrogen bonding patterns are a dominant factor in determining different types of protein secondary structures. In addition to these common stabilizing forces, disulfide bridges, which are covalent bonds between the sulfur atoms of the cysteine residue, are also important in maintaining some protein structures. For certain types of proteins that contain metal ions as prosthetic groups, non-covalent interactions between amino acid residues and the metal ions may play an important structural role. One classical example is the insulin hormone, where it is important in functionalization of matured molecules for it to perform its endogenous hormonal role. Through a cell biology lens, the study of gene expression is tightly linked to our understanding of proteins. While primarily concerned with protein molecules that act on DNA and RNA sequences, such as transcription factors and histones, the study of gene expression also focuses on where in the cell expression is modulated. In fact, the modulation of gene expression can occur in the nucleus, the cytoplasm, or even at the cell membrane, due to the impact of proteins on RNA in those cellular subregions. Ultimately, results from these kinds of studies have fundamental relevance, from the basic understanding of normal cell function, such as cell differentiation, growth, and division, to informing radically new approaches for treating disease. In fact, Some human diseases can arise simply from a defect in a protein's three-dimensional structure. Through the study of gene expression and proteins, it is easy to see how minute changes at the molecular level have a reverberating impact